Okay, so I'd now like to welcome Trisha Almazi up to the podium. So Dr. Trisha Almazi received her PhD in cancer biology in 2014 from the University of Sydney, Australia, and joined the Respiratory Technology Group in 2021. She's currently on a 50-50 split between the Woolcock Institute of Medical Research and a lead scientist at Ab Initio Pharma. The research interest in translational research, drug delivery, drug characterization, chemical proteomics, cancer, and chronic obstructive respiratory disease. Trish is an active member of the ECRE committee and chair of Work Health and Safety Committee, and has presented her work at several conferences, some of her work's also been translated into a marketing product. Today, Trish is going to talk to us about a novel thermosensitive in situ hydrogel formulation for sustained intranasal drug delivery. Thank you.
Thank you. So I'd like to start by thanking the organising committee for the opportunity to present our study on the novel formulation for the intranasal drug delivery. So this study was a collaborative effort between the Woolcock Institute of Medical Research, Tetratherix, and Ab Initio Pharma, where I'm the project lead. So a little bit about the current polymers that are used in nasal sprays. So thickening polymers are used currently in nasal spray formulations in order to increase viscosity of the nasal formulation so it actually stays inside the nasal cavity. And current polymers in use are Avicil and HPMC, which does the job of increasing the viscosity. However, on the flip side, it's difficult to aerosolize the high viscous solutions because with the current nasal spray pumps, it's difficult to aerosolize and particularly get inside the inner parts of the nasal cavity. On the other hand, with low viscosity solutions, there's a lot of nasal dripping, as well as wastage and low bioavailability. So our solution to this problem is to have a formulation with a novel mucoadherent and thermoresponsive polymer called tetramatrix. So what is tetramatrix? Well, tetramatrix is a novel, proprietary, and patented polymer, which is composed of 4 key monomers that you see up here. So one that, the first monomer that introduces its water solubility effects, another that, that gives it its thermoresponsive properties, and then we have something that can add the adhesiveness, so it can be mucoadhesive. And lastly, it composes of a functional group, which can be used for controlled release of drug if necessary. And together, it forms a structure similar to this. Now, I'd like to draw your attention to this video here, where we have a formulation with a nasal spray. And I just wanted to demonstrate how it can actually, using this polymer in a nasal spray, it can actually be sprayed quite effectively. And the surface that you see there is at body temperature. So as soon as it hits the body temperature, you can see that it starts to form a hydrogel, or it gelifies, and then it doesn't drip off. So that's I guess, the novel aspect of this polymer. So how can we use this polymer to improve bioavailability? So the hypothesis is, if you have the polymer, see up here, mixed with a therapeutic of choice, it could be mRNA, vaccines, antibody, or a small molecule drug, put it in an aqueous solution, such as that one that can be used in a nasal spray, and then spray it into the nasal cavity, where as soon as it hits the nasal cavity and at body temperature, it can form the hydrogel and thus be captured within that gel. Then, slowly, the therapeutic of choice can diffuse out of the gel and become available in the nasal cavity. So the study question we had is, can this TP polymer be utilized in a nasal spray formulation for sustained drug release? So the first thing we wanted to test is the sprayability of this formulation by using different concentrations. And the way we did this is to look at the spray characteristics. So the picture on the left here that you see is an experiment conducted on a flat surface with the VP7 Apter nasal sprays, and the surface is coated with a water detecting paste. And so the area of purple that you see here is essentially the droplet deposition on that surface. And what we observe is that with increasing amounts of the polymer, you can see that the distribution of the droplets is becoming less even. Additionally, we also observe that it also decreases the area. So the higher the concentration of the polymer, the significantly lesser area that it's covering. However, the ovality of the sprays are not affected, which is great. So now we actually use the control sample and the highest concentration sample, which is the TP140, to see how this affects the spray size or the particle sizes. So we did a spray tech analysis using the control and the TP140 samples, and we can see that it significantly increases the droplet sizes. So that may be having an effect on how it's depositing on the surface of that, on the flat surface. But the key question is this going to affect the spray when it's getting sprayed inside a nasal cavity? So the next series of experiments that we did involved using a nasal cast, a commercially available nasal cast. And here you can see the control sample, so it has no TP, no polymer in it, at 0 minutes when we first spray it, and up to 20 minutes. And at 20 minutes, you can see a significant amount of a nasal dripping, indicated by the red arrow over there. But then when we create formulations with the TP, you can see that the nasal dripping is completely gone. However, with the TP50 and the TP100, you see some dripping at the back of the throat, indicated by the red arrow. TP140, however, does not show any nasal dripping or any throat dripping whatsoever. So for the further studies, we took that TP140 formulation, and additionally, the area of the coverage that you see in that nasal cavity is comparable to that of the control, which is why we chose this to move forward with the next set of experiments. So the next thing we asked is, can this TP140 formulation have any cytotoxic effect on the nasal epithelial cells? And we did the study for about 24 hours of exposure. Here on the left, you can see in green the live cell staining. Here in red is the dead cell staining. Here is the nuclei, and this is the merged image. Qualitatively speaking, you cannot see any differences in the control and the TP140 formulations. Additionally, we did a quantitative study with a viability assay, and we've also shown that there is no significant decreases in viability compared to the positive control for cell death. So the last question we would ask ourselves is, can the TP140 formulation enable a sustained release of a drug? And in this case, we used ibuprofen as a case study. And so we used the transwell plates, which is a dual-chambered plate, which has an apical chamber and a basal chamber. In the basal chamber, we had PBS, and in the apical chamber, we deposited the TP140 formulation with the ibuprofen here in red. After an initial phase of burst release, we observed that 98% of the ibuprofen was captured in that hydrogel. So the next thing is, can the ibuprofen be released from the gel? And so we took samples from the basal chamber over a course of time, and we have observed that, yes, there is a sustained release of the ibuprofen, and it is in two phases, where in the first 48 hours, it is a faster rate of release at 44 nanograms per minute, and then it goes on to a much slower phase of release, up to 140 hours. And by 140 hours, we already have 83% of the ibuprofen released into the basal chamber. And if we zoom in closer to an area that is more physiologically relevant, which is 2 hours, you can see that by that stage, 8% of the ibuprofen was already released. We also wanted to see if interacting with the TP140 has any effect on the ibuprofen's performance. So ibuprofen is an anti-inflammatory drug, so we used a TNF-alpha model of inflammation, and we observed that compared to the raw ibuprofen sample, the one that interacted with the TP140 had no differences in decreasing the inflammation. So, in summary, we've characterized this novel polymer for nasal spray. It can be sprayed effectively by the VP7 Apter nasal spray, covering up to 94% of the nasal cavity within 20 minutes. It can rapidly form in situ hydrogel, which results in no visible runoff from the nasal cavity. It does not exhibit any cytotoxic effects for up to 24 hours on the nasal epithelial cells. And it can effectively capture and demonstrate sustained release of ibuprofen. So in short, it is an exciting polymer that can be used in nasal spray formulations, or can be thought to be used in the nasal spray formulation. And I just want to do the acknowledgements. So without these people, this study would not be possible. So everyone from the Wilcock Institute of Medical Research, Ab initio Pharma, and Tetratherics. Thank you.
Thank you, Trisha. Great presentation. Okay, folks, any questions for Trisha, please? One at the back, please.
Hi, Paul Hardman from Broughton. Yes, this is really fantastic. As someone who uses nasal sprays for hay fever, the nasal drip, etc., is annoying, to say the least. And clearly a very unique formulation. With that in mind, did the analysis using the spray tech, etc., pose any particular challenges?
So we just saw an increase in droplet sizes at DV50 and DV90, not at DV10. Did you want to know about the other formulations?
No, it was more around the actual technical challenges from the analysis. Did it clog up the machine?
Yeah, okay. So, well, it did not. But the trick is to actually keep it very, very cold, because at the cooler temperatures, it's very nice. It's very easy. It's a very easy to use formulation. It's sprayed very easily. So I guess the experiments there, we had to keep it very, very cold and then do the spray right after, and then it doesn't really have any issues.
Great, thank you.
We've got one through from Slido. Is the polymerization reversible? If not, how to make sure it doesn't polymerize during storage?
Reversible in terms of temperature, yes. So if we're talking about just having the formulation out at room temperature or close to body temperature, and then it polymerizes and forms a hydrogel, if you can put it back into a colder temperature, yes, it's reversible.
Okay.
And the second part was?
I think that was it. So that implies cold storage of the product.
Yes.
Okay, good.
We've got a question from the back.
Ben Forbes, King College London. You showed your cytotoxicity data.
Yes.
I don't think you said what concentration you used, so obviously the dose...
So it was the TP140 formulation, so the highest concentration.
Okay. I guess that concentration per... was that representative of the kind of concentrations you'll be generating for that surface area of nose, is one question. And related, I guess, so it's not cytotoxic, but biocompatibility more generally.
Yes.
Do you have any plans to extend those studies? Yeah,
so that's an excellent question. So this polymer was actually initially made for bone healing. So it's actually already been used in clinical trials. I think it was dental, what was it called? Yes, so for the dental implants, and it's all been through clinical trials and has no adverse events known. So it's actually safe for humans to use.
Okay. Another question down here.
Hi, Trisha. Excellent presentation. Sheetal from Aston Particle Technologies. So there are some polymers already available, like poloxamer, and also how different is your polymer? And second question is, I think you're going to cover the olfactory neuron area as well. So have you thought of toxicity to the olfactory neurons, and is there any brain absorption or something?
Definitely. So I think that what we need to do is extend the studies further. So we haven't done any sort of cytotoxic effect on nerves or neural cells, and if that is something that is that would be further down the line, depending on how the project, I guess, moves forward. And what was your first question?
How different is your polymer?
Polymer from others. Do you mean in terms of chemical structure or performance?
Yes, like I used poloxamer during my studies, so how better it is?
So I guess the novelty of this polymer is its thermoresponsiveness, and basically it does not increase in the viscosity until it hits the body temperature. So I guess that's a novelty for this polymer.
Okay, thank you. I think we'll stop there in the interest of time. Thank you again, Trisha.
Thank you.